See, I want to see how it coming. Currently live, setting up, waiting for YouTube live. Then redirecting to YouTube. For online classes, we need effective network as well as what? System. System should be well configured. Should be my system is used least configuration. Weak and flimsy system.
Good morning, sir.
Good morning, Aman Basha, Dikshida, Sai Bharat, Jai. You students, why this? You switch on your videos. Here we are still 10 students. Who is there? I don't know why you people are avoiding. Do you feel any in sometimes if you are not in a position, so you are not
Good morning, sir.
means you are not comfortable. Good morning, Anjay. What happened for last two sessions?
Sir, yesterday I was out of the station, sir, so I can't unable to join, sir.
You didn't join last two sessions, I think.
No, sir. Last one section only, sir. Yesterday also, last one section only I was not joined, sir.
Dhananjaya Rao, tell me.
Yeah, sir. From today I will be regular,
Out of
sir.
Okay. This is a kind of online class we are conducting. We need to finish the entire session. Sorry, entire classes. I don't see. So from yours, you students, this video, you see what is going on in your state. Even my mobile network is but outdated configuration connecting with the video. Students also connect. Definitely is possible. If you think it is a good school, so you good and connect. Don't underestimate your personality, your whatever you are. So, so today,
Sir, your voice is breaking, sir.
today I have connected with the live. Voice is breaking. Amman Basha is sitting in jungle. Ikshida, Ashida, I think that will Suganya is not correct. Students for today's session. So actually, just that book having different structure of into just I open the book in my system. One Basha,
Sir,
having so Zoom is connected or not? Clearly.
sir, I can't hear properly, sir.
Sir, your voice is breaking, sir. Continuously. I think poor network connection, sir.
That's what I am telling guys. Today, no, just I live streaming the YouTube. Just I tried and error basis. That's what I'm taking directly to. What sound is coming? Background noise. Manbash. Supanya. So, Dhananjay, did you see the screen?
Sir, it's
Dhananjay,
just it's loading, sir.
yeah, loading. Yeah, what?
Now it's visible, sir.
Yes. So,
Sir, sir,
please, you need your, yes. Okay, now it's clear. Okay, students. So, this is a press card book. The book is having for sorry, 7 evenings. I posted the first book in stuff. What today? Why it is called why it is called prokaryotic? The use of the term prokaryotic comes here. Then second, bacteria are but share some common features. See here they say about what is shape, arrangement, the size of the bacteria. See here maybe very classic. I mean there is some high definition images. See this is a cockeye. See first one is this is a very good images we have. See, this is cockeye. Chain chain. One more type of cockeye cluster form, blue color, looks very clear. Staphylococcus aureus. And here it is a rats. Bacillus mega. Rats in chain. So so it is seen by the name is color enhanced scanning electron microscopy 4800. So what we have discussed yesterday is what we they are telling about various first heading. Second heading, it says about plasma membrane. Plasma membrane is very important. I mean that is controls the what enters and leaves the cell. That's what yesterday I said. Is cell wall alone not controls. Cell wall protects the shape, shape and various other. But plasma membrane controls what enters and leaves the cell. So is a data structure. See, Dhananjay, did you see the structure? Is he able to see? Dhananjay, Sai Bharat.
Yes, sir.
Okay, this is clear now. It's visible.
Visible, sir.
Okay, thanks. So that's section 3.3. Yeah, you see the number of chapter 3.3 clearly explains about various what is plasma membrane and what is structure, how it controls the enters transport of materials. I mean molecules. So it's like a eukaryotic transport system alone. See the structure. It's very color illustration. Mosaic model. We know very well it is a fluid mosaic model. It's a bacterial membrane structure. So as usual, they have like proteins, then embedded transport proteins, integral membrane proteins. It's a connection between hydro say so no need mention about fluid model DNA, I mean plasma membrane structure. This is first repulsion electron transmission. That means electron they established membrane. There is a lot of information about it. This diagram shows the integral membrane proteins coating in a lipid bilayer. You know this lipid bilayer structure. That integrated protein is coating between. Different proteins are purple color. It is associated loosely with inner membrane surface. See everything very clearly. It's indicated. Small spheres represent the hydrophilic ends of membrane phospholipid, and the wiggly tails are the hydrophobic fatty acid chain. So this is a detail about how the lipid bilayer structure are arranged molecule as per the molecule structure of phospholipid. It is end set of hydrophilic end and the non-polar tail. This is the great portion is having ethanolamine. It is connected by phosphodiester bridging. Then glycerol long chain. So this long chain fat is called the hydrophobic acid chain. So it is you need to write something about phospholipid in fluid mosaic model of membrane. Say hydrophobic proteins. And you can also go there. Bacterial plasma membranes contain lipids not found in eukaryotic membranes. They have some special products. That's what it deals. Bacterial membranes are similar to eukaryotic membranes in that they are lipid bilayers or many of their antibody lipids and phospholipids. this is one difference. Oponites. These are steroids are synthesized from the same precursors of oponites are something unique for, I think, only for prokaryotes. Like steroids of eukaryotic membrane. Eukaryotic membrane is having some other cholesterol or yes, cholesterol, bacterio, this for this is for prokaryotes. B is prokaryotes. A for A structure having cholesterol is for eukaryotes. Then now it controls the movement of so bacteria use main mechanisms to bring nutrients into the cell. That's what movement of nutrients molecules between in and out of the bacteria. How it enables the synthesis of ATP as well. If you read it, you can understand everything. If you read line by line, definitely it will be no problem for you. Today I will share the book. From college I will share and after the college level. Now everyone will open the book. Simultaneously we'll discuss effectively. Yes, we'll get some. So the mechanism is involved is passive diffusion. There are many different transport mechanisms are involved in the transport of nutrients between in and out of the bacteria. Passive diffusion one, precipitated diffusion, primary and secondary active transport, and group translocation. So 4 mechanisms there are discussed in, for the subheadings. So first one is passive diffusion. Passive diffusion, there is no ATP, I think so. Passive diffusion, also called diffusion, simple diffusion, the process by which molecules move from the region of higher concentration to lower concentration. That is, molecules move down the concentration gradient. Based on concentration gradient, it moves from one side to another side. I think there is no energy required. There is no energy required for the movement of molecules by this mechanism, passive diffusion. So this is that's why the many substances cannot freely diffuse into a cell. However, some gases easily crosses the plasma membrane by passive diffusion. Oxygen, carbon dioxide is example. Oxygen, carbon dioxide freely diffuses from between the membrane by this passive diffusion. And the water also moves across membrane by passive diffusion. The point is there is a concentration gradient should be difference. The concentration, sorry, what is it? Concentration gradient drives the diffusion. So whenever concentration gradient fluctuates, based on that fluctuation, it moves. The carbon, oxygen, carbon dioxide, water, and larger molecules must enter the cell by other mode, other mechanism. That's what they are telling. So next is a transport passive. Sorry. So next is a fluid diffusion. Just you write some definition like this is a structure of bacteria. So we need to write the structure. draw the structure of the bacteria and the cell membrane, and also fine. But I think these mechanisms and all more than. So anyhow, just you what I'm telling is no and like some definitions about the what are the modes of transportation. So first is passive diffusion or simple diffusion, where small molecules only diffusion. But during precipitated diffusion, substances move across the plasma membrane with the assistance of transport proteins. They are either channels or carriers. So this is a see the structure. It will be very quickly understand if you see the structure. So there is some transport proteins which is bound the membrane and either act as channels or carriers. So here model of precipitated diffusion, there is no energy input. Molecules continue to enter only as long as their concentration is greater on the outside. So it is a like of diffusion. It is a diffusion, but this diffusion is precipitated by some protein that is called transport proteins. But normal type there is no such proteins. For simple diffusion, no proteins involved. Simply they are moving, crossing based on that constant gradient. But here there is one protein. Transport proteins are involved too. That is a precipitating the movement. See outside still inside, outside inside. There is more concentration. What is that? The spherical shape is called more molecules. Concentration more in outside. So it is binds with the transport proteins and enters to the inside. So simple logic. There is membrane. These are regarding membrane transport. Sorry, third one is a transport, active transport. First simple diffusion, precipitated diffusion. Third mechanism is active transport. Active transport is a there is some other there is further there primary and secondary transporter. See, this is a active transport. We know very well there is a ATP coupled with the active transport. See the structure, figure 3.13 ABC transporter function. So it very clearly shows the mechanism. Here, after binding of the AB transporter. So the blue colored integrated protein is called ABC transporter protein. So solute binds, once it binds the protein, so it will facilitate. This is a ATP drives the movement of solute across membrane. So this inside is cytoplasm, outside is a periplasm. This is periplasm. Once the solute binds protein, ABC protein, surface there is having some binding protein. So solute binding protein attaches to transporter and releases solute. Energy released by hydrolysis of ATP, which drives the movement of solute across the membrane. So the solute is yellow color, like see there is a yellow color mix called just image it is a solute. Once it binds to the transport protein, ABC transporter, what happens inside cytoplasm, there is ATP is a hydrolyzed to ADP and phosphate. So which enables the just like lock and key. So enables the movement or opens the transport of protein. So the point is active transport is coupled with the ATP. So ATP is necessary for the transport of fluids between membrane. Substrate binding protein free in cytoplasm, 3%. Other substrate binding protein are associated with the plasma membrane. Okay. So here they are telling what they are doing, primary and secondary. I don't know. Primary transport the energy provided ATP hydrolysis to move substances against the concentration gradient without modifying them. Primary active transporters are uniporters. That is, they move a single molecule across the membrane. ATP binding cassette transporters are important primary active transporter. So this model is a ABC transporter is a primary transporter, which is transport single molecule across the membrane. Single molecule hence it is uniporters. So secondary porter is a secondary active transport couples the potential energy of ion gradient. Uniporter, importer, antiporter. So secondary active transport couples with the potential energy of ion gradients to transport the substance without modifying them. So ion channel is a mediates the secondary active transport. Secondary active transporters are also called co-transporters. So co-transporter these two, importer and the antiporter. We have seen in many biochemical reactions. So these are importer, antiporter. Antiporter means that the molecules moves opposite direction. One goes inside, one comes inside. Sorry. Antiporter. There is two direction. So importer means both solute and that active transport comes inside. See, this is a structure. This is a diagram. It clearly says. Importer is active. These transport it has two types. Importer and data active. Importer means the substance move in the same direction. Antiporter means opposite direction. The diagrams are figures are more than sufficient to understand the concept. Then you read a bit more. So next is a group transformation. That is a fourth type of transfer of molecules. So group transportation. See, this is structure. Figure is explains about what is group transportation. So what is group transportation? The transportation of vehicles transfer from one part to B. So the distinct characteristics of group transportation, the molecule is simply modified as it is brought to the cell. So the change modify the structure. I think so. Modify the structure. See here, if you follow the structure, you will understand. PEP becomes the pyruvate. Then that pyruvate become PEP is indirectly enters to the cell. See, phosphorinolipyruvate, PTS are illustrated. The following compounds are involved in the system: PEP, enzyme 1, EI, low molecular weight stable protein, HPR, or enzyme 2, attached to enzyme B, the mental transport systems. So glucose is enters to the cell with the help of these kind of reactions. Glucose and mannitol. Glucose 6-phosphate and mannitol 1-phosphate. Read clearly, you can understand. Next is I will share today and tomorrow will see what is the silver structure. You can also open simultaneously in your system or system. That's what. So far, 7 only connected. Same 7 students. One year. Boys, wow, the connected. 2 boys and one more girl.
Bhargavi, sir.
Bhargavi. Bhargavi is not active. Okay, students. Thanks for joining. This is tomorrow.